Hello, welcome back. This next mini lesson is kind of the preview before we get to the physiology of how neurons actually function. We have to set the stage for what kind of environment neurons are in. And this is true for actually all cells, but we're going to focus primarily on neurons because that's the chapter that we're in. So we have these things called transmembrane potentials that basically indicates there's a difference in charge, like an electrical ionic charge, between the inside of the cell and the outside of the cell. That's it. So it's not equal. There's a difference. And so by 211, we called that like a concentration gradient, a difference between an amount of ions or sugar on one side of the membrane compared to the other. So it's kind of the same thing, but we're focusing on charges like ions, positive or negative ions, are different between the inside and the outside of the cell. And then we're going to take a look at how some ion channels can change those transmembrane potentials. And the big picture, if you want to, because we're going to get into some details of how action potentials work, so always kind of put it in the context of action potentials. These nerve impulses are the product of just changing the charge relative from the inside to the outside. And the unique properties of these membranes allow for that signal to be propagated or physically move down a membrane. We already saw a little bit of this when we talked about the muscle physiology, when we had the sarcolemma got activated at the neuromuscular junction, and we said the action potential moved down the sarcolemma, down into the T-tubules. That's exactly what we're going to be talking about. We're just going to now identify what's actually happening at the membrane to allow that action potential to travel along a phospholipid bilayer. So we have to kind of understand some basic characteristics about what we call resting potential. So in the rest of these physiological mini lessons, I'm going to show you this navigation picture. It's from an older edition. You can see all the way back from 2009. So I don't think it's in the newer editions, but I really like it because it sets up kind of what point we are at when we go through all of these processes. So in this video, we're going to focus on what we call the resting potential. What are transmembrane potentials, and what is it about them that makes a cell at rest? Okay. Then we're going to move on into a graded potential. So a stimulus shows up and it produces what we call a graded or local potential. And then that graded or local potential may or may not produce that action potential that runs along the axon. And then once you have an action potential, it will trigger what happens at the synapse. And then we'll see what that postsynaptic cell does. So there's like 5 parts of all of these membrane processes. This video is going to focus primarily on setting the stage for that resting potential. We have to understand that before we can change it to produce any of the other things that we see in the neuron. All right, so to understand the resting potential, we have to understand what we call this transmembrane potential, which is basically the resting potential. So if we'll notice, this was representing a plasma membrane. So if you remember, plasma membranes are a bilayer of phospholipids. I'm not going to draw the whole thing across, but just to remind you, remember it's amphipathic molecules. We have hydrophilic heads towards the watery cytoplasm and the watery outside environment. We have the hydrophobic tails inside. They prevent the transfer of charged particles through the phospholipid bilayer. So it is impermeable to ions. If we want anything to move through that is polar or large in size, we have to have leak channels. So here they're showing a leak channel of potassium. Potassiums are the orange plus signs. There's more potassium on the inside, so it wants to go out following its concentration gradient. I noticed earlier that in this leak channel here, it's going in the wrong direction. So that is bad. Bad arrow. Sodium is going to be coming in because we have a higher concentration of sodium on the outside. Sodium will be coming in through a leak channel. So that's just 2 ways that ions can travel across a membrane following their concentration gradient. But we also have to remember we have ATP-powered active transport. So hopefully you remember the sodium-potassium pump pumps things against their concentration gradient. So with the power of ATP, we can pump 2 potassiums in for 3 sodiums out. Okay? So the the relationship between the leak channels and the sodium potassium pump is as they work normally, they're maintaining homeostasis. Well, what's homeostasis? Homeostasis is a higher sodium concentration outside of the cell and a higher potassium concentration inside of the cell. That's normal. That's what we want to maintain. And what that ends up with is it allows us to have more positive charges on the outside of the cell and more negative charges on the inside of the cell, right? Didn't I just say transmembrane potentials or gradients are as a difference in charge? So the nature of cells with proteins and DNA and all these large negatively charged things inside of the cell make the cell at rest 70 millivolts more negative compared to the inside. So from this point compared to this point, from inside to outside, there's a measurement of negative 70 millivolts difference, and it's telling you that the inside is more negative than the outside, and that's perfectly normal. We do not want equilibrium. Equilibrium means no work can get done. There's no difference to take advantage of. So this is our resting potential, our transmembrane potential. Okay, so this is our rest. So to get a better understanding of how that transmembrane potential is regulated and then what happens when things kind of start opening up is we're going to take a specific look at the potassium and the sodium gradients. Okay, so in this upper left hand box here, this is a cell at rest. It's at resting potential, right? Negative 70 millivolts is our resting potential. And it's just basically going to show you what potassium would like to do if channels were, if the membrane was freely permeable to potassium. So for the chemical gradient, right? So the chemical gradient is a difference between quantity of chemicals. So those little orange circle plus signs, there's way more potassium on the inside of the cell than on the outside. So potassium, if channels open, would want to go out of the cell. That's the chemical gradient. But because it's charged, and I set up the difference in charges of the membrane, the outside is more positive, the inside is more negative. Because potassium is a positively charged ion, its electrical gradient wants it to go towards the negative charge because we know opposites attract. So if we're just following the electrical gradient, potassium would move into the cell. But we have to see the net electrochemical gradient. So we have a whole big push to push potassium outside of the cell and a little bit less push to bring it in because of the electrical charge attraction. So if you sum those together and you find the net movement of potassium, we would have a net, excuse me, loss of potassium outside of the cell. So if the membrane became permeable, potassium would leave the cell. And that's actually what they're trying to show you here in the second picture, is that with the membrane completely permeable, which this would never happen completely, is that if we opened the membrane, potassium would leave so much that look what happens to that transmembrane potential. We would lose enough potassium to make it go from a negative 70 at rest to even more negative because we're losing positive charges. So if we lose positive charges from the inside of the cell, it's going to make the cell more negative. So at negative 90 millivolts, potassium is at an equilibrium. Okay? We reach that for a tiny, tiny little split millisecond in action potentials, but really this is not a normal scenario for cells to be in at a negative 90 millivolts. For neurons. Other cells have different kind of values. All right, so now let's take a look at the sodium resting potential. Okay, so again, here we're at negative 70 millivolts. We have way more potassium on the outside of the cell than on the inside. So if we were to follow the chemical gradient, if membrane channels were open, sodium is going to rush into the cell. If we look at the electrical gradient, because sodium is also positively charged, it is attracted to the negatively charged inside of the cell. So if we combine that to our net electrochemical gradient, we have a huge influx of sodium coming into the cell because both of those gradients are in the same direction. The electrical gradient wants to move sodium in, The chemical gradient wants to move sodium in. So we add those together, we get this huge change or this huge influx of sodium if the membrane becomes permeable. And so when we see that happening, look at how much difference that transmembrane potential becomes. We start at -70. If the membrane were to become completely permeable to sodium and reach sodium equilibrium, so much sodium would have come into the cell that we have changed that charge all the way to +66. Now, this is something that we don't ever reach in a normal physiological process. So if we were to let potassium become equalized, it would become more negative than normal because we're losing positive charges. If we let sodium equalize, channels open, membranes permeable, we would gain so much sodium that we would totally flip that transmembrane potential into being more positive on the inside compared to the outside. Okay? So those drives, if you will, of potassium and sodium help us understand what's going to happen when we actually do start opening and closing ion channels during graded inaction potentials. Okay, the last slide here is how do we open and close channels? Because we know that's the only way we're going to get these ions to move inside, into and out of the cell. Because they're ions, they have to go through a channel. So this first one, the chemically gated channel, you've seen that already. We saw that at the neuromuscular junction in the motor end plate. It was acetylcholine came in. So they show that as a picture. So in resting state, the gate is closed. Okay, no sodium can come in. They're just using sodium as an example, but it's true for all these different kinds of chemically gated channels. If the receptor or the ligand comes in, so ACH is a ligand, it's going to bind into the receptor site. That's going to physically change that ion channel. It's going to open, and we now get sodium flooding into the cell. So that's one way that we can open channels and allow sodium or other ions into or out of the cell, called a chemically gated channel. It allows for like a lock and key. So here's the ACH is the key. It's going to unlock the lock that's going to open the channel. The middle one, which is like super important for action potentials, this is called a voltage gated channel. So its key, if you will, is not a chemical like in the first example, it's change in charge. So for this particular one, this is a sodium voltage gated channel. This is specific for the sodium ones that we're going to see. So at this particular channel, it's got two gates. We have an activation gate, which is on the outside of the membrane, and we have an inactivation gate, which is on the inside. At rest, so if I'm going to draw my gates, here's my channel, and here's my gates. Okay, so at -70 millivolts, we are going to have the activation gate closed, which means sodium cannot pass through, and the inactivation gate is open. But it doesn't matter because the activation gate is closed. You just need one closed to prevent ions from moving. If we change that, if somehow we change that transmembrane potential to -60, that's the magic number, if you will. This is a number that we call threshold. So we're changing that environment right around this gated channel, that when that happens, that activation gate opens. When that activation gate opens, the membrane is permeable, sodium is going to rush in. And we know that sodium is going to rush in until it reaches positive 66, right? So sodium's rushing in, rushing in, rushing in. We're going -60, -50, -40, 0, +10, +20, +30. And when we get to +30 millivolts on the positive side, that's the magic key for the inactivation gate to close. Okay? So at +30, the gate closes. And so that's how we can open and close a voltage gated channel, is these little doors will open and close based on the charge environment around it. Okay. And then lastly, we have mechanically gated channels. We just mentioned these here. We're really not going to see them too much in the rest of this chapter. We'll see them in the future when we take a look at touch receptors. But this is the gate is closed until it physically gets distorted and opened by pressure being applied. Sodium will rush in, we can activate the cell, pressure goes away, gate closes, the cell becomes not stimulated anymore. So we're going to focus primarily on the chemically gated channels and voltage gated channels for the rest of the physiology of this lecture. Okay, so I will see you next time for graded potentials. Bye.